First of all, welcome to the first Global Open Genetic Engineering Competition. So I'm Kaylee Rallick. I'm a PhD student at Rice University and one of the founders. So we'll have our official opening ceremony in a little bit, but today I'm excited to offer and announce our first guest speaker, which is Dr. Ann Meyer. She is an associate professor at the University of Rochester, where her research focuses on using quantitative techniques in the fields of biochemistry, microbiology, and biophysics to study structural dynamics, macromolecular interactions, and physiological responses of organisms to environmental stressors. She uses the tools of synthetic biology to engineer novel functions into microorganisms, with a particular focus on the production of improved tunable biomaterials and the development of tools for 3D patterning of bacteria. So please welcome Dr. Meyer.
Thank you so much for this invitation. It's really exciting to be a part of the first round of Gogec, and I hope that it continues on for future years. I think it's a really great initiative. Let me pull up my slides. All right, is that looking all right? People always ask that, even though it always is. All right, so yeah, it's great to be here. I'll tell you a little bit about the more synthetic biology oriented projects in my lab. So the overall goal of kind of the symbio half of my lab is, can we use biotechnology and synthetic biology approaches to turn bacteria into factories for making materials, right? So people are probably more familiar with bacteria as factories for making small molecules like insulin or antibiotics or something like that. But I think that there's also, vast untapped potential for bacteria to make, the structure of the built environment around us. And can we do it in a way that's environmentally friendly and replaces traditional and more wasteful approaches? So, I think the bacteria are amazing candidates to be the next generation of materials production factories. Unlike brick and mortar factories, bacteria factories replicate themselves. They're really cheap and easy to duplicate and make more of. I love the fact that they have evolved mechanisms to catalyze really sophisticated chemical reactions, but typically in a really environmentally friendly way, right? So typically, aqueous solution, water-based solution, close to room temperature, close to standard pressure. They don't usually make or require toxic compounds to do their chemical reactions. If you consider unlike, for example, pouring cement, bacteria can deposit their materials on bumpy or cracked surfaces, vertical surfaces, in outer space, microgravity. And these factories are easy to transport from place to place to make the materials on site. And thanks to developments people in this room today, they are increasingly programmable for giving the potential to make even more sophisticated tunable materials in the future. So let's start with the first inspiration for my lab was, and it doesn't sound obvious, but it was the problem of cement, right? So cement, everyone interacts with it every day, probably more or less. I'd be curious to hear people that don't actually, but cement is popular for a reason. It's really easy just to mix up and pour into simple shapes. One reason why it's really popular is that it's got high compressive strength and high stiffness, right? That means you can squeeze it really hard before it breaks. And stiffness is a material science term. Stiffness means that if you poke it really hard, it's not going to change shape. It doesn't deform when you poke it, right? But there's a lot of downsides as well to cement, compared to other building materials. It's got high density, it's very heavy. Before it solidifies, it's very caustic, it's got very high pH, so stay away from wet cement for that reason. You can't really do fine spatial patterns with it. As far as the mechanical properties, right? So I mentioned it's got high stiffness, right? It doesn't deform easily, but the toughness is very low, right? So toughness is if you're trying to bend it, how much force does it take before it breaks? So cement breaks really fast, it has low toughness. This is why you often see, cracks on the sidewalk, right? In the road, especially where I live. Besides the material properties that I think can be improved, the environmental sustainability is terrible of cement, right? So the cement industry alone is responsible for 8% of global carbon dioxide emissions, which is outrageous. And there's additional 4% also when you consider making concrete as well. So my lab was wondering, can we make something inspired by cement that has better mechanical properties, much more environmentally friendly? So we took our kind of everyday concrete inspiration and combined it with natural inspiration, which was nacre. So what is nacre? Nacre is also known as mother of pearl, or like pearl mother in a lot of languages. It's the inner layer of mollusk shells or bivalve shells. It's also what makes up pearls. Nacre is an amazing material because it is 95% calcium carbonate. So it's basically 95% cement. But so that gives it like the high stiffness of cement, like I was talking about. However, in comparison to cement, nacre's fracture toughness can be up to 1,000-fold better than that of cement. So it's this amazing material that combines toughness and stiffness, which usually you don't get in the same material. If you think about, I don't know, I have all these like starch analogies, right? If you think about a cracker, right? Like a cracker is just what you eat, or a crisp or whatever. It's very stiff, right? But it's not tough. But then if you compare it to like a slice of bread, right? A slice of bread is very tough, but it's not stiff, like you indent it really easily. So just, nacre is an interesting example of a natural material that's a rare example of combining high toughness and high stiffness in the same material. Because of its amazing mechanical properties, lots of groups are really interested in trying to produce versions of nacre, in the laboratory. However, most of these approaches require like really expensive equipment that you run in your fume hood, and they typically produce lots of organic waste that has to be disposed of afterwards. So my lab is interested to use biological techniques to make nacre-inspired materials that are easier and cheaper than chemical approaches, and also more sustainable environmentally. All right, so here's what we have to make. So this is what you see if you take just the shell of a blue mussel that you might buy in the grocery store, the fish market. If you cut it in half and look at the cross section, this is what it looks like. So I mentioned it's mostly made of cement. So this is what we see in this electron microscopy image. So the calcium carbonate crystals are organized into platelets, so kind of like long, thin, like plates of cement. And you can't see it in this image, but the plates of cement are basically glued together by sticky biopolymers. That's the other 5% of nacre. So you get this kind of like sandwich, of like calcium carbonate, sticky stuff, calcium carbonate, sticky stuff that builds up the shell of an oyster or a mussel or a clam. And this, really intricate nanostructure is at the heart of why it's so fracture tough, right? So, if you take the structure, if you poke on it, calcium carbonate gives it good stiffness. If you try to bend it and break it that way, the fact that it's got these alternating layers means that if a crack starts to form in the bottom, it can't propagate through the layer of biopolymers, right? And it has to have a very long fracture propagation length before it gets all the way to the top of the material, Or if you have a little bit of force that breaks a couple of the platelets, that can be okay because the rest of the platelets didn't get fractured and they're going to stay intact. And there's other mechanisms as well that keeps it really tough. All right, so in our lab, when we want to make a nacre-inspired material, this is what we're trying to make. So we need to a way to use bacteria to make layers of cement, and we need to use bacteria to make some sticky, hopefully, like malleable type of polymer. So can you make cement with bacteria? The answer is yes. So this was, people in the field before me have discovered this amazing bacteria that just lives out in the wild and mostly in puddles in people's backyards, called Spore Sarcina pasteurei. It has really high levels of an enzyme called urease. What this means is that if you're growing the bacteria and you add some urea in there, it's a common byproduct of animal life, it will break the urea down into ammonia that turns the pH, makes the pH go higher and higher. When you get to pH 9.5 or 10, then you have spontaneous crystallization of calcium carbonate, right? So this is a picture of what happens if my student Dominic Schmieden took a culture of this bacteria, in the bottom of it put a small just glass platelet, like a microscope slide, added some urea and waited overnight. until the reaction, go to completion and see what happens. We pull out the piece of glass and look at just the top view under a microscope. You see it's got this beautiful layer of calcium carbonate of cement covering the whole material. So this is how we can make cement with bacteria, really easy, just overnight on the benchtop. All right, so we also need now some sort of sticky polymer layer, like to stick together our layers of cement. For this, we're using another wild type bacteria called Bacillus licheniformis. These are found on bird feathers. I've never known why, but that's where they like to live. These bacteria are really interesting because as part of their natural lifestyle, they, outside of the cells, they make and excrete massive amounts of this polymer called polygamma glutamate. So it's glutamate like, the amino acid, just strung together in these long polymer chains with gamma linkages instead of amino acid linkages. That's not that important. But that means it's really easy. Again, you just grow a culture of this bacillus bacteria, for a few hours, let it make all of the polymer. You call it PGA for short. Once it's made some polymer, if you add ethanol or you add copper or just something to precipitate out the polymer, and you can kind of like stir around and pick up this giant gloopy ball of gum, and that's your polymer. So this picture is what you get. My student Nadine Bonhertz had just taken like a 20 mil culture of bacteria and got this giant mass of polymer out of it. So it makes a lot of polymer and it's really easy to actually, you don't even just extract it, you just collect it. Okay, so here's our two starting materials we want to use to make our hopefully Nacre-like layered material. So our general approach is you take a thin piece of plastic. This is the type we use called PMMA in the little blue line there. You put your PMMA plastic in the one beaker that has the calcium carbonate precipitating bacteria in it. You let it precipitate the calcium carbonate, make a layer of cement. You take out your plastic and you put it in a different beaker that has the collected polymer, the PGA polymer. Let that settle and make a layer. And we go back and forth to build up our material. So typically, like Dominic and Eva Spies in the project would do this, 20 times, get a nice thick structure. So after we do that, after we do this reaction cycle 20 times, we can take the material, again, snap it in half, look at the cross section under SEM microscopy, and here's an example of what we would see. So Dominic got this amazing picture of the cross section of our material where you see these beautiful layers going all the way across the cross section. It looks just, very similar to the layered material structure you see in natural Nacre. So that was very exciting for us. So, but we don't want it just to only, look like Nacre. We want it to have the really good mechanical properties too. So Ava on the project had a great material science background. So she did a lot of mechanical characterization of the properties of our material. Here's one example of the kind of test we did. This is a test looking at fracture toughness. So, how far can you bend it before it starts to crack or fracture? So this is called a 3-point bending experiment. Basically, there's two platforms on which you balance your material. That's like the 2 points of the 3-point bending experiment. And there's a probe that comes from the top at a certain defined force. And then you can see, the deformation behavior and cracking behavior of your material over time as the probe, interacts with your material. So on the left, we're looking at our layered, kind of looks like Nacre material. On the right, we're comparing it to, this is a sample treated the same as a layered Nacre, but we never added any of the polymer. So basically it's just 20 rounds of laying down bacterial cement. So it looks like just like a bulk chunk of cement. This is our non-layered control sample. All right, so as the experiment goes on, this is the kind of results that we can see. So they behave very differently in the 3-point bending experiment. So on the right-hand side, where we just have the bulk calcium carbonate, what we see is that, small cracks form very early on, like lower extensions, lower forces, and they quickly propagate. So they, once they form, it splits right through the material and the whole sample just cracks in half, right? On the left-hand side for our layered material, we also get cracks, right? So micro cracks, like very small cracks, do form. We can see that afterwards by looking at the sample under the microscope. But in the layer structure, these cracks don't propagate, right? So they form, but they don't get any bigger. So the overall material stays intact, which is just what we were hoping for. And after many different types of approaches and experiments, this is the kind of summary of the properties that we see. So our bacterial nacre, our layer structure is here in green, and we're comparing it in this kind of white oval to the properties of natural nacre. So there's, lots of species of bivalves make nacre, so they have kind of a range of toughnesses. What we can see is that on the y-axis, we're looking at the toughness of the material, and our nacre, the bacterial nacre, has a toughness equal to or better than many different types of natural nacre, which is very exciting. And we can still improve on the stiffness, but overall, it's quite stiff compared to typical bacterially produced materials. All right, so to summarize this first project, we can use only bacteria to make two different kinds of structural type of materials. So we can make the tough layered composite material. And I don't know if it's interesting for anyone to have a brittle material, but in case it is, we can make kind of a more brittle, just like bulk cement kind of material. We can make it in macroscopic amounts, large quantities. Even though our nacre is not as stiff, we're like, 10-fold less stiff than natural nacre, it's still the stiffest artificial nacre ever produced, which I think is a great milestone. It's exciting that it's as tough as natural nacre, because that's the famous property that everyone wants to replicate. It's exciting that it's highly extensible. You can bend it very far without it being brittle. And I love that this entire process, it's not releasing carbon into the environment like industrial calcium carbonate production. It's actually fixing carbon into the material, which makes it vastly more eco-friendly, I think, than traditional cement industry approaches. So that was kind of our first foray into making a structural material using bacteria. And there's a lot of directions we want to go in after this point. So, one thing that we were hoping to improve on in our, upcoming techniques was, can we make more nacre faster, right? So, I love how tough our material is, but it takes a long time to go, from bath to bath, bacteria culture to bacteria culture to build up your tiny little nanolayers, right? So can we make more faster? So for this approach, we were inspired by a different kind of mollusk, which is the gastropod, so snails, right? So similar to oysters and clams and mussels, which are bivalves, snails, which are gastropods, also make shells, right? They're also, nice and tough and stiff, but they have a totally different approach to building up their shells. So oysters and mollusks, they make their shells the same way we made our first round of nacre, which is layer by layer, slowly over time, they get, bigger and bigger. Snails take kind of a quick and dirty approach, if you want to call it that. So it's a two-step approach, which is kind of exciting for optimizing your time spent making it. So, there's two components in nacre. There's the polymers and the calcium carbonate. And so the first step is they make a bunch of polymers, like a thick matrix of polymers, kind of cross-linked together. And then the second step is they'll crystallize calcium carbonate through it. And you can see in this picture on the right, this is kind of a snail shell caught like in the process of being made. You can see these kind of like, not to be overly Western about it, but kind of Christmas tree structures, kind of as, or, pine tree structures, like as the crystals are forming. Right, so we were really attracted by the idea that instead of like spending 20 steps to make a 20-layer very thin material, could we have 2 steps to make a thick material that was also kind of a similar crystallized polymer kind of material? So for this approach, we wanted to try a different polymer we thought would be more suitable for this approach. So, to make the calcium carbonate platelets, we're using the same bacteria that's really good at mineralizing calcium carbonate. But we have a different polymer, which we thought we would try bacterial cellulose, made by a species of bacteria called Gluconacidobacter hansenii. I usually just say G. hansenii because it's a lot easier. All right, so what's our overall approach? Let me just talk briefly about what this bacteria is like. So I don't know if any of you ever drink kombucha, but kombucha is something where, it's a living beverage, if you want to call it that. And some of the living components of kombucha is some bacteria that makes bacterial cellulose, right? So this picture is similar to what you'll see if you just take your kombucha and just leave it on your kitchen countertop for 2 weeks, which is that the bacteria that make this cellulose is very hydrophobic, so it goes to the top of your liquid. and then it'll just get thicker and thicker. And so you get this like super dense, really densely cross-linked hydrophobic matrix meshwork of bacterial cellulose. And, with very little effort on like the human's part, right? You just start your culture, sit it on the bench top, come back 2 weeks later, and then you peel off. It's kind of like a giant chicken breast, like floating on your kombucha or floating on your bacterial culture media, right? It's ready to go, right? It's like a big, thick layer of cellulose, ready to go. So how do you turn this into something similar to a snail shell? So the first image here is our student Kui Yu holding this is like a giant piece of cellulose that he got from taking like a roasting, like a baking pan full of bacterial culture that made a roasting pan layer, like roasting pan size sheet of cellulose. But the cellulose is so hydrophobic that you can't mineralize it with calcium carbonate. We had, we kind of bounced off this project several times where if you just take this cellulose, put it with the bacteria that makes the calcium carbonate crystals, it's so hydrophobic that, the solution that's making the crystals doesn't penetrate into the cellulose. So you end up with just your chicken breast full of cellulose with crystals on the outside and on all the different outside surfaces, but not inside. So that's not what we're going for. So, we love taking low-tech, cheap approaches when we can. So basically, Kui just takes the giant platter of cellulose, puts it in the blender, blends it up for a few minutes, and then we can mix it with our bacteria that make the calcium carbonate crystals, which this approach is so obvious. I wish we thought of it a year earlier. It's okay. Then once the bacteria forms all the calcium carbonate crystals, we can boil the solution to inactivate the bacteria, put it in whatever shape mold we want, and let it kind of spontaneously reassemble its final structure. So the final material we get has, we were really excited with the material properties that we got. So we got these materials that were very resistant to mechanical forces. So here I'm showing 2 samples of materials. The top one is one where we took the cellulose and mineralized it with chemical, kind of CO2 releasing processes. And the bottom one is when we use the bacteria to make the calcium carbonate minerals. And then Kui took it to the balcony in the building and dropped it off the balcony 3 times. So when you drop it from 10 meters high, the bacterial material down at the bottom doesn't noticeably change shape. It's not affected by those forces at all. But the one that was chemically mineralized just gets pulverized to a pile of dust. So it's really tough as far as that goes. And it's really rigid, right? So I mentioned that it's moldable, right? So after you make your kind of mineralized cellulose slurry and you sterilize it and you mold it, Kui can make a variety of different shapes. He made kind of a pen holder, and he made this kind of thin-walled bowl. A thin-walled bowl was rigid enough that he could turn it upside down and stand on it with his entire weight, and it didn't crack or fall apart. So yeah, I love how it's not only tough but also very rigid. And then the moldability is really interesting too. So here's some examples of, this is all the exact same sample that Kui would mold and remold and remold. So first of all, he put it in this kind of pretty mold that was like a snail shell shape. And then once it forms its shape, and you have your nice rigid tough sample, you can take your material, soak it in water, put it back in the blender, put it back in a different mold, and then it forms a different rigid tough shape. And this is the same sample kind of taken through a cycle of all different shapes. So I like how it's really flexible, and I think end of life considerations are important for products to think about what happens to your product when you're done with it. And this is a product that doesn't have to have a defined end of life. You can just turn it into the next thing that you want to use, assuming it's something that's rigid and tough, which a lot of things are that you want to use. So I'm really excited about our different versions of Nacre we can make using bacteria. There's a lot of, I think, interesting examples of ways this could be useful to human society. As one example, since our materials are not only tough, but very lightweight compared to typical structural materials, I think they're really interesting for components of airplanes or ships or rockets, something where you want to minimize on your fuel costs by having your vehicle be as lightweight as possible. we saw that our material was very resistant to forces, like impact forces, which I think makes it interesting for, bike helmet, shielding materials, cell phone cases, anything where when you, impact your sample, you want it to stay intact. I think an interesting idea is that since our materials are all very biocompatible, right? So all we use are, biopolymers made by organisms. Some of them are edible. So far, I think they're all edible, actually. And then calcium carbonate, just similar to your tooth and bone material already, right? Could this be some sort of like orthopedic implant material? So, for example, right now, if you break your bone and you have to have like a pin implanted to stabilize it while it heals, typically you have to go back for a second surgery to take the pin out because it's not long-term compatible with your physiology. So couldn't this be, could this be something where you could, make the pin or the bone graft of the shape you want, embed it in your bone, but then not have to have the second surgery? I think that would be a huge step forward. Oh yeah, we had this interesting collaboration with a makeup company who's interested in the light reflecting properties of our nacre. Nacre, I didn't talk about this much today, but besides having great mechanical properties, is also very beautiful because reflects the light in these kind of iridescent ways due to its high refractive index and this kind of interesting nanostructure. So this has caught the attention of companies that want to basically take our like beautiful nacre, grind it up, and then put it in makeup so it reflects the light and conceals your pores. One thing I like about a material is that since everything can be made on site, could this be a way to make our future moon or Mars habitats, right? So if you're like, if you want to go and live or work temporarily on the moon or on Mars, personally, here's my like hot take on the topic. Personally, I don't think it's going to be cost effective to send up all of your building materials on your spaceship. That's going to take a ton of rocket fuel that can be saved and used elsewhere. I think you should save the rocket fuel for people. And then if you send up on your spaceship just a small tube of bacteria, once you get to the moon, once you get to Mars, you can propagate your bacteria and then make your building materials on site, which I think would be a great approach. Okay, yeah, so for the future, it's a lot of stuff we want to tackle. We'd love to, I think we have good toughness at this point. I'd love to improve the stiffness through various engineered approaches. That's already, underway. If we can make our layers thinner, which seems to be working so far with genetic engineered control over the urease enzyme that makes the crystallization reaction happen, we seem to be able to get thinner layers, which in theory should lead to both tougher and more iridescent materials. Of course, we'd love to have something where we can like tune the ratio of polymer to mineral and get tunable flexibility. This would be like, if you think about eggshells, right? So lots of animals make eggs, and they're all basically made of calcium carbonate and polymers mixed together. But bird eggs are really stiff. They don't change shape easily because they have a lot of mineral and a little bit of polymer. But reptile eggs, snake eggs and turtle eggs have less calcium carbonate and more polymer, is the ratio of those two components. So they're much more flexible. So I think it'd be really interesting to use a synbio approach to be able to tune the mineralization reaction to get tunable flexibility in our final product. We definitely want to continue on this road of speeding up production. I think that's really promising for the future. We're doing a bunch of, starting to do tests for kind of real world properties. So does this stand up to environmental forces for kind of long-term use? And one thing I think would be really fun would be, can we make our material to have more of a shape, right? So I already showed you can make kind of the bowl and the kind of pen holder structure, but those are kind of not really precise spatial patterns, right? So can we have like much more control over the three-dimensional spatial patterning of our materials that we make? So this is like a whole other part of the lab. All right, so our challenge on patterning bacterially produced materials is, how do you pattern the bacteria, right? So people grow bacteria, they grow it like in a shaking liquid flask where everything's mixed together, or you can grow it on the surface of some solid media where, over time, the bacteria colony gets like wider and wider, and then your spatial patterning gets lower and lower resolution, right? So how can you grow bacteria in a certain defined shape so that their materials that they produce end up being also in a certain defined shape? So this is a big part of the labs working on new technologies to do this. So this was, maybe you guys have heard of the iGEM competition, probably you have. This was something that the iGEM team that I ran in 2015 when I got my use of work in TU Delft tackled. So this team, I was super excited with their project. They developed the first, to my knowledge, the first ever bioprinter designed to print bacteria. It was the cheapest bioprinter ever made at the time, around 350 euros. And it was really, such a fun project because it was fully do-it-yourself. Because you can see, like, our students are holding it in the picture here. Because it was fully made from the ground up using Connect's construction toys. It's such a colorful, like, fun toy. I think it's great for educational purposes. So if people want to make Connect 3D printer yourselves, we published the full instructions for this at ACS and Bio together with some of the iGEM students. And here we are after having won the grand prize that year in iGEM. So yeah, so I think this is a really good approach for kind of educationally getting kids excited about 3D bioprinting. And just the general idea of 3D printing of bacteria, I thought was super compelling. So thanks to these students' amazing idea, I've continued working on it in my lab. So we've had several iterations of improving the 3D printer. The kind of version 3.0 is the one we published most recently, but version 4.0 is hopefully coming out next year at some point. All right, so how does our upgraded 3D printer work? So here's the most recently published version. So we still want to keep our inexpensive do-it-yourself ethos. I don't know if anyone's ever priced out what it costs to buy those 3D printers for making organoids or whatever, but those can cost $100,000 or more. And I don't think that's accessible for the typical laboratory, right? So this printer, we're keeping it under $500. So, it's still quite cheap compared to, commercial 3D printers for biological systems. Compared to our toy printer made of Connects, this one is much more precise, which is, I think, great for taking the application further. So the general idea is the following. So your bacteria, you can call it bioink, is in a syringe. The syringe is kind of in the back in this kind of red apparatus. It's a liquid culture of bacteria, and it's in a syringe pump, the red thing in the back. So the syringe pump, what it does is it kind of just squeezes the plunger of your syringe at like a specific speed that you program in. And then the bioink, here just red, just so you can see it, is squeezed out the syringe through some tubing over to the print head. So this is the print head of, this is just like literally the cheapest conventional 3D printer that we could find. It's like some sort of like, $250 3D printer that we bought. And it was designed to print plastic, like PLA plastic. It kind of gets melted and extruded and then dropped down onto the printing surface. So, my students on this project, Benjamin, later in this case, just took off the extruder, took off the heating element, and replaced it with just the input tubing here. So what we get from this 3D printer is the precise positioning of the printhead over time, right? And then the syringe pump is making sure that our bacteria is being squeezed out at the end of the tube, so it's being deposited on our printing surface at precise locations over time. So you have good automated control over the speed of the pump and the printhead motion. But how do we control the chemistry? We don't want our liquid culture to stay liquid, right? So my students developed a special bacteria bioink inspired by eukaryotic cell printing techniques. So basically, how it's been working is we print onto a surface that contains calcium ions. In the past, usually we've been printing on top of, agar plates, just the kind of typical bacterial growth media. So it contains food for the bacteria as well as calcium ions. Our bioink is a liquid culture of bacteria. We mix in alginate. Alginate is this, monomer that can form a polymer. Then the printhead goes by, deposits our bioink. The little black threads are the alginate. Then after printing, the deposited bioink will interact with the calcium ions on our LV plate there. So the calcium will, diffuse up, interact with the bioink, and calcium induces the alginate to polymerize, right? So now it forms a hydrogel, kind of like, agar, jello, whatever. So now our liquid bioink is solid. And the bacteria are trapped inside the alginate matrix because the pore size is smaller compared to the size of the bacteria. So it's more or less like trapped in place over time. The great thing about a hydrogel is that it's mostly water. It's like 95-ish% water, which means that, first of all, the nutrients in the agar can continue to diffuse in and feed the bacteria after printing. But also you can print subsequent layers. It's really a 3D printing approach, because if you print a second layer on top of the first layer, the calcium will continue to diffuse to polymerize the second layer, and so on and so forth. So it's easy to print. We've printed, up to 20 layers using this approach. More is possible if you just spritz it with calcium, in between. There we go. All right. So here's some examples of just kind of fun shapes that Ava and Kui wanted to print, just to show that we can print arbitrary shapes using our 3D printer. And you can change how fast the print head is going, how fast the pump is pumping, to change the width of the stripes of bio ink that you're depositing. And with this approach, and also you can change how much calcium there is on the surface, because the more calcium makes it polymerize faster, there's less spread after you print it. So, with tuning these parameters, it's easy to get down to sub-millimeter printing resolution. So a single stripe would be maybe like 0.3 millimeters in width. So, one thing we'd like to achieve with 3D printing is, can we deposit different things in different places in our structure that we're building? So if we had different kinds of bacteria, could we deposit them in different places within our structure? So to test that, we had an experiment where Dominic had bacteria engineered to express either yellow or a blue fluorescent protein, and he printed them, one as the first layer, and then one as the second layer. We can take confocal microscopy and zoom in at the interface between the two layers, and we see, quite good separation, right? So, more or less, they stay to their respective layers. You can see a little bit of mixing just right at the interface, but it's maybe only like 5 micrometers in z-axis height. In comparison, the bottom image is when we don't have the bio ink chemistry, we don't have the alginate, the calcium chloride, we just have, we're depositing liquid culture, and you can see everything just fully mixed. Okay. So, when we print our bacteria, we want them to not just look pretty and sit there, but do something. So once we print our bacteria, we want them to be metabolically active and healthy enough to make something of our choice, of our interest. So here's just kind of a toy example where Benjamin Lehner was printing a cube of bio ink containing bacteria carrying a plasmid to make RFP, red fluorescent protein. This is something where the bio ink just had the bacteria, but we printed onto a substrate that had the inducer to induce RFP production, such that it was not able to be induced until after it was printed, but we hoped the inducer would diffuse up and induce production. That seemed to work fantastic, thanks, because the inducers, IPTG is very small, so it diffuses well through our bio ink. So we were able to get really good production of RFP over time. Which gave us hope that we could make other elements of our choice, other bacterial products of our choice in our 3D printed structures. Right. So what do we want to make with bacteria? I'm going to share the 2 most recent examples from my lab, because they just came out like a few months ago. So after we had a couple of papers of printing various things with bacteria, we wanted to see, can we print other different types of microbes, right? So we're interested in the idea of, could we print not bacteria for the first time, but could we print microalgae, right? And with the idea that since microalgae is photosynthetic, could we make a living material that's also photosynthetic? So what is microalgae? I'd never used it before. We started this project a few years ago. So we use we use a microalgae called Cloudimonas reinhardtii. It's like a really popular microalgae for lab usage. It's, microalgae is an interesting beast because it's kind of partway between animals and plants, if I can call it that way. So it's single celled, like bacteria, so it's easy to print as far as that goes. It's eukaryotic, so it's a lot of features similar to animal cells, but it's photosynthetic, like plant cells, by the fact that it has like flagella that help it swim around. So it's got interesting features of both kingdoms. But for us, the most important thing was it is very small. And so hopefully we can print it evenly and have it be trapped in our alginate matrix, and it's photosynthetic. So can you make a photosynthetic material? So here's an example of what happens when we just do our normal printing process to print a microalgae in our like normal alginate biochemistry. We can make something, we can make like a little photosynthetic t-shirt structure, but it's not robust physically, right? So just kind of any sort of like physical perturbation and it crumbles, right? So I didn't really like the mechanical properties of this material because it was really fragile. But then Kui had this great idea, and I already mentioned the amazing properties of bacterial cellulose. Could we kind of combine that together with our microalgae? Right, so I mentioned how he can like grow his cellulose in this giant like roasting pan, right? So he grows a big roasting pan full of bacterial cellulose, peels it off, and when it comes off, it's a really thick layer of material. But then if you just let it sit and air dry, it becomes kind of something between like paper or linen fabric. It becomes this like kind of canvasy kind of material. So it's flexible, it's thinner, and it's really tough. Right, so what if we use that as our printing material? Would that change the overall mechanical properties of what we make? So so what do we get when we print onto these different materials? So one thing I thought was really fun was that since our microbes can feed themselves using only sunlight, our living material can also feed themselves using only sunlight. So on the right side here is when we print onto like an outer plate with some nutrients for the microalgae, and we can either just grow them in the dark, so their only food is from the food that we give them on the growth medium, and they grow a little bit, or we can grow them with food, and we have kind of a light dark cycle where we like have some grow lights, like your greenhouses would use, just turning on and off during the day to make a light dark cycle. And in that case, the material gets really green. So you can see the microbes, microalgae are growing, dividing, they're photosynthesizing, and they're turning green over time, right? And even if we don't give it any food, if we don't give it the carbon supplement in the media, we only give it the light from our grow light, it still gets green over time, right? So this material feeds itself and sustains itself without us having to feed it, which is great for like long-term sustainability of this material. And I love the fact that it really seems to thrive. Like we weren't sure if microalgae would like to live in this, bio ink that we printed in. But, if you come back and look at your material over time after like days or even 4 weeks, every time we looked at it, it got greener and greener, right? And as just a visual depiction, we also had assays to look at, cell density and chlorophyll content, and those also increase a lot over time, right? So they seem to really not just be surviving, but really dividing, photosynthesizing, making chlorophyll. So it's really a living material. How's the time doing? Need to wrap it up soon. Okay, we also scale it up. So, we can like print our living material all over the entire roasting pan, a sheet full of cellulose to make like a really large, piece of material. And the cellulose gives it really great properties, right? So we can take our printed microalgae, once it grows up, it's slightly more robust, and we can basically just like peel it off one piece of cellulose and put it on the other one where it sticks instantly. So it's kind of like a reusable sticker or something like that. And it's really nice and robust, right? So we can fold it and twist it and crush it. So Kui and Srikanth Balsubrahmanian had like a really fun day kind of trying to destroy the material every way they could think of. But, if you unfold, untwist, uncrush your sample, it looks the same afterwards. It doesn't crumble. So it's, it got really good physical robustness, I think, due to the cellulose properties. And I love the fact that it is, really living material. So here's an example of Kui and Srikanth. They printed, a certain shape with the microalgae, let it grow up to be nice and lush and green. Then you can kind of depolymerize the alginate, turn it back to liquid, grow it up a little more use that ink to print 4 times as many things as you printed the first time, so printing the same thing 4 times, and that all grows out to be equally lush and green as the first thing. So, think of this as kind of like sourdough starter, right? Like this is something where you can make a material, hopefully deploy it out in the world, give a chunk of it to your friend, and they can make their own starter culture to make their own future living materials. So here's this amazing picture that one of my former adjunct students made to illustrate our project, just to show that we can make these photosynthetic living materials that are physically robust, self-sufficient, they feed themselves after you print them, and they're, I think easily able to be shared and distributed, kind of like, bread starter, which I think has kind of very like science fiction-y applications in, living clothing, photosynthetic skin, but more like practical applications, I think, include, membranes for filtration that are really sustainable and, keep themselves viable, or artificial leaves, right? Could this be a way to harness solar energy similar to the leaves on a plant, but in such a way you can deploy it again, like in space, like very high, high efficiency ways without having to worry about a plant that has to have, a trunk and roots and stems and stuff that don't contribute to the energy fixation. So potentially this could be like a really easy way to store solar energy that's like as easy as just like leaves gathering the sun on the plants in your backyard. Okay, so just quickly I wanted to give like another short example of something more bacteria focused that we also have been working on recently, which is can we tackle the problem of biofilms with our 3D printing approach? So biofilms are when bacteria, which, you think of bacteria as like a single-celled species, and kind of they are, but then in many different environmental conditions, they choose to live in a multicellular community where like lots of different bacteria live together in this community, kind of stuck together by lots of excreted extracellular polymeric substances that make a thick matrix that stick them down to a surface so they kind of live together. And in this lifestyle, biofilm living bacteria are very robust. So they're really hard to detach from the surface. They're really sticky. They're really hard to kill. So bacteria that used to be susceptible to antibiotics or bleach or whatever in a biofilm might be resistant. So these, really resilient biofilms can be good, right? So they're often kind of a passive part of water treatment plants to purify drinking water. Their biofilms are used to extract the majority of copper and copper mine tailings. But for human health, they're not fantastic. Dental plaque that causes cavities, that is a biofilm, which is really gross to think about living on your teeth. And some people think that the majority of infectious bacteria-caused infections are caused by bacteria living in a biofilm lifestyle, which is tricky because this is the lifestyle in which they are not easy to eradicate using typical small molecule antibiotics. So my very first, my adjunct students that did the very first 3D printer were interested in this application. And they went to talk to a local toothpaste manufacturing company, like a Dutch toothpaste company. And so they grow these model biofilms in their laboratories. They can test like how well does their toothpaste get rid of, dental plaque biofilms. But, they told us that from week to week, the biofilms weren't really consistent. So they couldn't really be certain, like, is their toothpaste getting better or is it just a different biofilm formulation this week? So we thought this could be our 3D printer could be an interesting way to make, to reproduce the spatial patterning of biofilms in a reproducible engineered kind of approach. So what's our approach? So our idea is if you 3D print bacteria, we're in this case 3D printing E. coli that have been engineered to express a certain protein. So after printing, the inducer molecule diffuses in, induces production of the CHGA protein, which bacteria will naturally, E. coli will naturally export it, whereupon it will spontaneously form these long fibrils called curly fibers, if you've heard of curly fibers or CHGA fibers. So these fibers are the major protein, like proteinaceous component of the extracellular matrix of many types of biofilms. It's what sticks the bacteria together and it sticks bacteria down to a surface. So this is what we can engineer production of in our 3D printed microbes. So, we show that we can produce these curly fibers in our printed bacteria, but now, therefore, do they act like natural biofilms? Well, there's a couple of assays we can look at. One assay is, so natural biofilms, the extracellular matrix is really dense and hydrophobic, so oxygen doesn't penetrate. So the bacteria use up the oxygen and then doesn't come back in, it doesn't diffuse in from the outside. So you build up this like depletion of oxygen, lower depths of the biofilm. So Shrikant Balasubramanian, he would 3D print certain types of biofilms, incubate them for a week, let them become mature and make their curly fibers, and then like lower a probe through to test the oxygen concentration at different depths. Right. So what we're looking at here is like in black, if you just have some alginate matrix, there's like good oxygen diffusion in the whole structure. If we look in blue is Nissel. It's like a wild type biofilm forming species of E. coli, strain of E. coli, and you can see that as you go deeper and deeper, the oxygen drops off really fast. If we have in gray kind of our lab strain of E. coli that doesn't make these biofilm components, the oxygen drops off like much more slowly. But in orange is our engineered bacteria that makes the curly fibers, and that has the steepest drop off of all the different strains we tested. So we see really good oxygen depletion, similar or even better than our native biofilm species that we looked at. And then like the important thing for clinical application is, do we see the resistance to antibacterial treatments like you see in native biofilms? So for this, we had an experiment where we looked at, we 3D printed the biofilms, Shrikant cut a little sample, treated it with different things. In this case, we're looking at ethanol, which kills bacteria. Then we looked to see how many bacteria survive. So on the y-axis is how many bacteria survive per mil of printed sample. We see that they survive pretty well when there's no ethanol, right? But then as there's going from left to right, when there's more and more ethanol, there's look at the gray, the unengineered bacteria that doesn't survive very well in the higher ethanol concentrations. In blue, the native biofilm species survives quite well compared to in gray. And in orange, which is our engineered bacteria that makes the curly fibers, that also survives really well, almost as well as in blue, the native biofilm species. So I was really excited that we saw, two different major biofilm phenotypes, including good resistance to different antibacterial treatments in this engineered 3D printed system. All right, so to summarize, I think our 3D printing approach is really exciting because it's a really inexpensive 3D printer that we try to engineer it so that anyone, you don't have to be an engineer to make your own printer in your own lab. So if you want to make your own printer, we publish some methods articles, or please just email us if you want advice. I think this could be interesting for continuing this work on making standardized biofilms. This could be like a test bed biofilm on a chip approach for screening new antibacterial components that could actually kill biofilm lifestyle bacteria. That's so important for human health. And we've got a recent grant to see if we can use our 3D printer to kind of embed bacteria in or on plastics that are deployed in the environment so that maybe they wouldn't live forever in the environment, but the bacteria could break them down at end of life. We've had some collaborators in South Korea that are very interested in combining our technologies to see if we can make like robust devices for purifying water and water treatment plants that could be actually reproducible and definable. So in water treatment plants that use these biofilms have a big role in like sucking up the carbon, the nitrogen, the phosphorus as the water goes by, but the water source changes, like in Flint, Michigan, right? Like that can really strip off a lot of the stuff from the inside of the pipes and the filters of your water treatment plant, and it kills your biofilm. And then, typical water treatment plants, like no one knows what species were in the biofilm and you can't reproduce it. But could we use this approach so that you can reproduce it and replenish it whenever you need to? And I would love to, combine our 3D printing approach with our other materials production, right? So we're right now working on, can we 3D print our bacteria that make the nacre, to make nacre with any shape? We also have a paper where we showed we can use a different bacteria species to make graphene in solutions. Can make, 3D structures containing graphene. I think the artificial leaf approach is really fun. If we 3D print the bacteria that makes the cellulose, people in the field have thought this could be a way to make the personalized skin grafts that like fit your personal shape. And like the fact that our materials are really alive means that we can make three-dimensionally patterned materials that respond to environmental cues. So could they respond to cues in the environment to change their toughness, to heal themselves, to detect contaminants and remove them, or even kind of report on your, human health to be a living biosensor. So yeah, I'll wrap up. So our lab can make living materials, like using 3D printed microbes, use bacteria to make structural materials like Nacre, conductive graphene materials. We're also using bacteria to make optical materials or space materials. And we're developing new approaches for molecular protein sequencing, which is also a very interesting topic. And I'm happy to answer any questions.
Okay, so if you guys have any questions, you can either just say it out loud, unmute yourself, or feel free to message me if you have questions as well.
So I had a question. So regarding the artificial leaves, do you or like with the sourdough starter type thing, I was wondering if there were any applications in like carbon fixation or anything like that you thought might make sense with that technology?
Yeah, I think it was interesting. I know that, I went to this Department of Energy workshop where they were interested in, some carbon fixation approaches require you to have like a tank of CO2 that you feed to your system, which that's very energy intensive to make the concentrated tank of CO2. This could be an approach where you're, like you mentioned, like you're fixing carbon, but in a very kind of passive way that doesn't require energy input, which I think is very interesting.
Cool. Yeah, that does sound interesting.
So I actually also had a quick question. So I know you said in like the future works for the bacteria Nacre, you're interested in tuning like the thinness of the layers and the ratio of the calcium carbonate to the biopolymers. But how do those properties currently compare for your bacterial Nacre to like the conventional Nacre, and what sort of parameters in this bacteria culture might you change to kind of tune those parameters?
Yeah, definitely. Yeah. So with this project, we're like just starting to get good data on it. The student gave lab meeting yesterday. It's very exciting. So our approach has been, instead of using our kind of environmental bacteria that can mineralize calcium carbonate, we're taking an E. coli that's been engineered by our collaborators this time, the Shubar lab in UC Boulder. And so they have an E. coli engineered to express the urease enzyme, either with like kind of strong or weaker expression strength. And then what we're seeing is that if we use these enzymes, these bacteria to make our Nacre, or layer by layer built up Nacre, that the one that has the fast urease, like the stronger urease expression. So, crystal growth is not always intuitive, right? So when you have faster urease activity, like, stronger expression of the urease enzyme, you actually get smaller crystals because crystals are weird. And this is interesting because the first results we have show that the cross-section of our Nacre seems to have thinner layers, which is very interesting because in theory, the more thin layers you have, the tougher your Nacre should be. And potentially also, so like the iridescence of Nacre is a function of the fact that natural Nacre, the layer height is, like 500 to 700 nanometers or so, which is, the same range as wavelength of visible light, like the kind of blue-green spectrum of visible light, or just, which means that, if we can get layers that are thinner in that range, which I think we're starting to accomplish, then that should maybe also be iridescent. And, it's not as kind of practical for structural applications, right? But there's a lot of people that contacted me after our paper came out that are interested in, can you buy natural Nacre and use it to make inlay on, like culturally, important drums or guitar inlays on the frets or whatever, right? But people want to make a version of that that's vegan if they can, like doesn't involve animals. And plus, if you have like a big thing you want to cover in Nacre or mother of pearl, you're kind of at the whim of how big is a seashell, what shape is a seashell, right? So if we can make something that was beautiful, like a seashell, but any size or shape and vegan, I think it's a big market for that, I think, as well, demand.
Okay. Well, I think we have time for maybe one more quick question if anyone has one. Okay. Well, then, if not, I will now pass it over to Shyam to introduce our next speaker.